Well, thank you all for coming back, and thanks to the morning speakers. And this was a great introduction, and we will carry on with the agenda. My name is Ivan Rusin. I'm from Texas A&M University, and I'm a member of the committee. And I was asked to pitch in relief of Dr. Zarbel, who has a lot of activities remaining today and tomorrow on this workshop, and he was the lead planner, so I want to thank him as well for leading the committee to an excellent lineup, and this has been a very interesting session thus far. Our continuation of this morning's session is more on the environmental impacts and impacts of chemicals and the interactions between chemicals and the microbes. And we have two outstanding speakers in this late morning session. Our first speaker is Dr. Kun Lu from University of Georgia, and he did his PhD work at University of North Carolina, Chapel Hill, and then his postdoc was at MIT. He's an excellent mass spectrometry person and a chemist, and it's very rewarding to see that he's now moving into the field of microbiome as well. So Kun, please take it.
So first of all, I would like to thank the committee for putting this exciting workshop together. So I'll be talking about the effect of metal exposure with the arsenic as a primary example. Today I'll be talking about three sub-titles. So the first one is a functional impact of exposure on the gut microbiome. The second thing is the relationship between the gut microbiome phenotype variation and the metabolism. The last one is the role of microbiome in the gender-specific toxicity. So a very quick introduction. We're going to use several slides. So gut microbiome has an important function, we all know that, particularly in the food digestion, metabolic processing, biotransformation of xenobiotics. And I want to point out the gut microbiome induces not only the local effect in the gut, the effect can reach to the distant organ in the liver and even brain. And the study already shows many diseases actually influenced by the gut microbiome shift. And the next example I would like to set is the gut microbiome community structure is associated with disease and particularly obesity in this case. And also the fecal transplant experiment confirmed the metabolic function change associated with gut microbiome community structure. So this actually is the basis of many studies look for the gut microbiome community change as the first endpoint. And also, I want to point out the gut flora regulated metabolites actually is also important. Okay, so this is an elegant study to show the TMAO trimethylamine oxide is a disease-causing metabolite, which is initially generated from the choline and trimethylamine from our diet, phosphatidylcholine. So that makes sense to look for the metabolites which can cause or promote the disease development. So consequently, we can do it using the next generation sequencing to study the 16S to do the microbiome community structure survey. And also we can move forward to do the metagenomics and metatranscriptomics using the RNA-seq to examine the functional impact of gut microbiome change. And also meanwhile, so we have a huge number of metabolites compound in the body. So particularly if you see the here, the microbiome actually contributes a significant amount of compound in our body, which offers the opportunity to using those metabolites as a surrogate to study the functional change of microbiome. So we used both targeted and untargeted metabolomic approach, both DC mass spec and LC mass spec based metabolomics profiling. We studied the metabolic change. Eventually, we reached the metabolic pathway or functional difference between the disease state and treatment. So the first question we try to ask, what's the functional impact of exposure on the gut microbiome? So the question, the fundamental is the gut microbiome can be easily modulated by different external factors, such as diet, drug, bacterial infection, and so on. So change is very easy. So what? So we have to check whether it's function change associated with the community structure change. And from the perspective of exposure, environmental exposure, so right now an increasing list of chemicals are reported to alter the gut microbiome of selected bacteria, which include metals, pesticide, PCB, and so on. And the list is quickly expanding now. So we use this workflow to explore the impact of chemical on the gut microbiome and its function. So first, we do the 16S mapping. Then meanwhile, we do the metabolomics profiling. So we're trying to establish a functional correlation between the gut microbiome change and metabolic perturbation. So the example here I'm giving is arsenic. So because arsenic is a chemical with a global health concern, it's not only a problem for some South Asian country, for the US it's also a problem because a lot of people is drinking the private well water, which is not regulated by any regulation agency. So it's estimated 20 million people is drinking the water with the arsenic level above the EPA standard. And arsenic has been linked with a number of diseases, including the cancer, and more recently with the diabetes and the cardiovascular disorder. And for arsenic as a mode of action, and the metabolism is also very complicated. I'm going to come here in a second. So we first treated the black 6 mice with arsenic. As you see here, that's the control. That is the arsenic treated animals. So the beta diversity of gut microbiome is separated very well, and they actually cluster in their own category. And we identify several Firmicutes bacterial family was significantly perturbed. Then the next, we do the metabolomics profiling. So first, we check the feces sample. We extract all the metabolites from the fecal pellets. The feces actually serve a better sample to assess the gut microbiome derived metabolites. So as you see, the hundreds of metabolites either up or down-regulated. And we also check the urine and the blood to see what's the impact in the host. And we identify quite a few metabolites. So I'll give you some examples. The first one is the indole derivatives. So this compound, 3-indole-propionic acid, that's a very unique compound. This gut flora generated compound. If you check this one in germ-free mice, you won't be able to detect this one. So now we see it's about 2-fold increase. And also this one, the indole-oxy, we see it's about 3-fold increase. Indole-oxy is starting from the indole. Indole actually is one of the major substrate bacteria can work with. So the microbiome regulated metabolite is suggest the bacteria, either bacteria or bacterial function was perturbed by arsenic. Another example I would like to use is isoflavone metabolites. So we identify several downregulated, and upregulated isoflavone metabolites. So it's well-established isoflavone metabolism. The gut bacteria is involved. In the human, some are good equal producers, some are not. So the change of those isoflavone metabolites actually could serve as a surrogate to assess the gut bacterial functional change. And in fact, we can generate a correlation plot using some typical gut flora regulated compound and change the gut microbiome family. So the blue one is a perfect positive association, and the red one is a perfect negative association. And this one to show some specific metabolites and specific bacterial family. So you see the indole lactic acid positive with B9 bacterial family and negative correlated with the B8 bacterial family, and so on for the diazoxin, for some bile acid species. So certainly, we can actually start from the microbiome change and assess the the function change using the metabolomics approach. So as Andrew mentioned this morning, so that's maybe a way, I mean, to get some functional readout using the microbiome to metabolism approach. It not only works for arsenic. Here is another example from the nicotine. Okay, so as you see here, so this one, so nicotine-induced the dose-dependent gut microbiome change. Correspondingly, you see the dose-dependent metabolism shift. And those metabolites actually correlated with some endpoint we accessed in the host. And next, I'm going to move on to the second question. So the relationship between the phenotype and the gut microbiome variation and the individual response. So the question is, so there are large individual variation in the gut microbiome composition. It's not only in the human, also in the animal, you're going to see the huge variation from animal to animal. So the individual response is a big deal in the toxicity study. So over the years, we have seen many factors contributed, such as genetics, such as nutrition. Then how about the microbiome? So then we decided to assess whether the gut microbiome related with some xenobiotics biotransformation difference. So first, we induced animal model with different gut microbiome. So how we do that? We're using two ways. The first is bacterial infection, which is a natural way to modulate the gut microbiome. The second one, so we use a gene knockout model to mimic the genetic background difference. Then we map the 16S profile, and we measure the arsenic species using the ICP mass spec. So as I mentioned, arsenic metabolism is very complicated. So this involves the methylation is monomethylated and dimethylated. And also they can be either trivalent or the pentavalent, okay? But the methylation is considered a detoxification pathway, okay? And this one, so dimethylated arsenic 5, that is a detoxified metabolite of inorganic arsenic. And also I want to point out that this one is the cell-lated arsenic. That's usually considered a gut flora generated compound because bacteria can put a cell group here. And also some bacteria actually is also involved in the methylation and reduction pathway over this cycle. So we can actually measure almost all the arsenic species using the SCP mass spec. So this one to show the bacteria-induced gut microbiome change. So in the during the bacterial infection, about 15% the gut bacteria family was perturbed. And correspondingly, we measure the arsenic species. The first one we checked is the dimethylated cell-lated arsenic, because that's gut flora generated compound. As you see here, at multiple time points, they are different between the control and the treated animal. And also you see the dimethylated arsenic 5 is actually reduced. And the inorganic arsenic 5 actually increase. That's suggesting in these animal models, gut microbiome shift inhibit the arsenic detoxification, okay, because the dimethylated arsenic 5 reduced. Similarly, so if you compare the gene knockout model and wild-type animals, so you see huge gut microbiome shift by single gene knockout. So, and you see they cluster very well. More than about 70 or 80% of gut bacterial family actually different between the wild type and the knockout animal model. And similarly, we measure the arsenic species. You see the dimethylated arsenic 5 reduced, and the inorganic arsenic 5 increased. And meanwhile, you see monomethylated arsenic 5 also increased. And if you calculate this ratio, so actually it's higher in the knockout animal model. And in fact, this ratio is commonly used as a biomarker in the human population study to assess the individual susceptibility or disease susceptibility after exposure to arsenic, because that tells you the biotransformation or the metabolism of arsenic in the human. Okay, so this animal model actually mirrors the situation happen in the human population. And we also did the correlation between the individual bacterial family and arsenic metabolite. So here is some correlation plot. So the last one, I'm going to touch on whether the gut microbiome contribute to the gender-specific toxicity and disease susceptibility. Because it's well known, I mean, some disease has gender-specific susceptibility. For example, for autism, male to female ratio about 4:1. For the Parkinson's disease, about 2:1 ratio. And in terms of the environmental toxicant, some neurotoxicant induce the gender-specific toxicity, particularly in male in some cases. So because there are huge difference between the male and female in terms of gut microbiome, so we hypothesize this may contribute to the gender-specific toxicity and disease susceptibility. So then we're using this workflow. So again, we map the 16S, and we do the metagenomics, and we do metabolomics. Then we put them together to check whether there is gender-specific effect by the exposure. So this figure you see that it's very good separation. The only factor here driving is gender. So this one is female, this is male. Okay, so the huge difference that's actually override any treatment. Okay, in this case. And also I want to point out that the gut microbiome is dynamic. So even it's after a few weeks for the animal, so it's not very young, I mean, for the animal, but still you see the dynamic change for here. So this one is the baseline, and here, the bottom one is after about 4 weeks. So you see significant shift over the time, okay? So that's actually raises the question. The baseline between the male and the female is different, and also they have the time-dependent shift over the time. So whether this arsenic can induce the gender-specific in terms of the microbiome change over time. So the answer is yes. So the female and the male, you see, they have very different pattern. So in terms of the effect on the trajectory of gut microbiome. So, and those are bacterial genus. And here is the female case, and this is the male case. And also, we actually further examine the functional metagenome. We also show the gender-specific pattern. So it seems that gender is really play a role in this type of response. So we actually also checked using other chemical to see whether that's chemical specific or it's kind of a general potential general mechanism. So in this case, we use the pesticide. So again, we see the huge gender specific change. And if you look at the metagenome, so in the male, you see about 6 functional category change, but in female, it's only about 2, and they are totally different. And this one is actually correlated with the metabolites measurement and other toxicological endpoint in the host. So we believe that the gut microbiome may play a role in the gender-specific toxicity or even in the disease susceptibility in the human population. So a very quick summary. Okay, so there is a functional interaction between the gut microbiome and the environmental exposure, and the gut microbiome may serve as a novel factor of individual susceptibility and gender selective toxicity of chemical. And also, I want to point out the challenge actually remains to characterize the functional impact of exposure-induced microbiome change in the host, because any measurement that you do, you actually combine the effect from your exposure and gut microbiome change. So it's actually challenging. And with all that, I would like to acknowledge a number of people and the funding agency for the financial support. And I would like to thank you for your attention and take any question you may have.
Again, if you're asking a question, please introduce yourself.
Hello, my name is Captain Blair Dancy from the U.S. Army Center for Environmental Health Research. Thank you for your talk. In your course of your sequencing studies of the arsenic exposed animals, did you consider, and this sort of relates to a question that was posed earlier in regards to the glucuronidases, did you look at mobile genetic elements or consider them as a potential source by which the microbiome might be reacting to the arsenic? Along those lines, I don't know if you might have any comment on the utility of perhaps like a probiotic approach that incorporated like bacteria that perhaps were optimized to metabolize arsenic, from its more, I guess, toxic forms like arsenite to some of the, the methylated versions to which you alluded. So sort of that approach and whether you think how practical that might be.
Yeah, so a good question. So on the first question, we haven't got there yet. Okay, so honestly, but we start to look at from the sequencing, the deep sequencing data using the metagenomics and the RNA-seq. And for the second question, I think that's a very good approach. So we are actually working on that using some food component to modulate the gut microbiome to see whether we can change the arsenic metabolite profile. So if it succeeds, I think that's all for a good intervention strategy to reduce the arsenic toxicity in the human population.
Hi, thank you. Jason Richardson, Northeast Ohio Medical University, and a member of the committee. Again, thank you for a very nice talk. Two related questions. First, when you were analyzing the males versus the females, where were the females in the estrus cycle, and does the microbiome change if you start analyzing that across the estrus cycle?
It's a great question, but unfortunately, we haven't looked at with that details. We just collect the microbiome at different time point. We usually do it every week to collect the feces sample. So we don't know yet, but that's really a great point to check that.
Great talk, Kun. Ann Summers, Georgia. The point I'd like to speak to was the suggestion that arsenite is less toxic, and that's not so. It's a different kind of toxicant. Arsenate, which is what is in the redox state 5, the body really wants to keep it in state 5, and that's why it hangs all these methyl groups on it. Arsenite is a thiol poison at arsenic 3. So you don't really want to have a whole lot more of that made. The bugs, in fact, use that reduction. Use the arsenite 3. They have a reductase actually that they reduce arsenate and then they pump it out as arsenite. So keep that in mind. It's not non-toxic. It's a different kind of toxicant.
Yeah, that's a good point.
The metals, with the metals, speciation is everything.
Okay, thank you.
Okay, so Helmut Zarva, quick question. So relating to what Jason asked you about the sex differences, which is very intriguing, did you look at overrectomized animals? And were you able to reconstitute, a male microbiome in overrectomized females or something like that? Did you change the susceptibility?
So see again, what's your question?
So did you look at overrectomized animals is the first question?
No, we haven't.
Okay, and did you try to take the animals that were female, for example, and give them a male microbiome and show that you actually changed the?
Yeah, that's a good question. That's actually on our plan to see whether we can actually switch that. And also, we are actually looking for what's the factor actually is driving the gender-specific difference in terms of microbiome.